Today we're going to be discussing decision-making and executive function. So clearly brains do decision-making. We make decisions, and our brains are responsible for doing that. We don't know all of the details, obviously, as far as decision-making goes. This is a complex process, and it's one that's tough to suss out. But we have learned a lot, and there's a lot of very interesting experiments that we're going to discuss today. So many animals appear to plan and decide. You can even see, if you've got a pet dog or a cat, like they make planning and decisions. They want to play, they want to go outside. They will let you know this. Humans, though, appear to be unmatched in their ability to respond flexibly and anticipate the outcome of behaviors. This is really the thing that sets humans apart from other animals. Planning and decision-making, it depends upon a bunch of things. Sensory input. You can't make a decision about what's going on in front of you without being able to sense what's going on. Remembered information. You have to make decisions about what has occurred, your prior experience, anticipation of if you make a decision, what will happen because of previous experience. Goals. The internal decision-making of what do I need at the moment? Am I hungry? Well, that's going to probably affect my planning and decision-making at the moment. Am I looking for sexual reproduction? One way to put it. That will also affect your planning and decision-making. And then, of course, predictions of what might happen. So using the sensory input, combining that with your remembered information, that and your prior experiences, obviously that's going to lead to an anticipation of what you expect to happen, weighing the odds of those things. All of this is information and processes that lead us to make decisions and make plans. The limited capability to study this issue in animal models limits to what we know so far about the brain, areas that regulate these behaviors. We can do some of these experiments with humans, and we will look at data in humans when we can look at activity related to fMRI. You can do some of this work in animals too, using fMRI or doing in vivo recordings. But the problem with doing this in animals is that you can't just ask them, hey, what were you thinking at this moment? They can't tell us that. We can do that with humans, but then we're limited in the kind of neural information that we can get. So you can see that because this is an internal process, this is something that we can only look at the outcomes of behavior. We can't really necessarily define what is happening in an animal's mind other than what their overall behavioral outcome was. So we have to suss these things out and then correlate that to what we see happening in humans. So we're going to discuss the major brain areas involved in thinking, planning, and decision making. So we're going to break down some of these areas. The prefrontal cortex is key to a lot of the decision-making, planning, and logic areas of the brain. We have subdivisions that we kind of have broken down to be functionally distinct. The dorsolateral prefrontal cortex and the ventrolateral prefrontal cortex are two areas of the cortex that we're going to be talking about. The insula is another important area. So this is an area, if you retract, that's what these things are. These are retractors. So you imagine a brain sitting in front of you, and you can pull away the temporal lobe. Behind that will be the insula. That's what that's called. And then on the medial side of the brain, we've got a number of important functional subdivisions. More of the ventral prefrontal cortex, but this is the ventral medial prefrontal cortex. The orbital frontal cortex is also key. The dorsal medial prefrontal cortex. The dorsal anterior cingulate cortex. This is the cingulate here. So dorsal anterior, so it's dorsal and it's anterior, to the posterior cingulate cortex, or the PCC. And as you might have figured, we have the reward areas of the brain, the caudate putamen, which is the striatum. So the basal ganglia figure into this as well. And we've kind of already gotten to that point because of the procedural memories that we have learned Obviously, that can be involved with thinking and planning and decision making, but we've already talked about that. Instead, we're going to be mostly focusing on the higher cortical areas and their various roles in decision making, planning, and logic. So humans and other large apes have more of their cortex dedicated to the prefrontal cortex. That's illustrated here. So here we've got a bunch of different mammalian brains. And you can see in the human brain, what we're showing here is the prefrontal cortex. And it's a good chunk of the cortex, like maybe a third. Chimpanzees, maybe it's about a quarter or so. And you can see in macaques, this is another primate, very smart primate. And it also has a relatively large prefrontal cortex. Squirrel monkeys are smaller than cats. You can see that their overall brain size is about the same, but by weight, squirrel monkeys are actually smaller. And they have a little bit more dedicated to the prefrontal cortex than cats do, which, to be honest, I've got a cat. I don't think he does a whole lot of planning, decision making, and logic beyond just sleeping and eating. And rats, they have prefrontal cortex, but it's also very tiny. And this is generally true for a lot of the other mammals. And you can measure out what this looks like. And there have been various studies that have done this, looking at the absolute size of the prefrontal cortex or the proportional size. So relative to the rest of the cortex. So absolute size, this is the red here. So these red dots is showing the absolute size of the prefrontal cortex. And this is log scale. So you have to kind of take that into account. So we're looking at millimeters cubed. So this is a volume, and we're looking at the volume of the brain that is prefrontal cortex. And gibbons are a type of ape. They're kind of the outgroup of the apes, which also includes chimpanzees, orangutans, and gorillas, and humans. That's all the apes. And then we have macaques, which are a very advanced monkey, but they are monkeys. One way you can tell the difference between a monkey and an ape is that apes don't have tails and monkeys do. So macaques, you can see their overall prefrontal cortex is smaller than all the apes. And gibbon is slightly bigger. They're also not much bigger in size than macaques. I think they are a little bit. And then we have orangutans, chimpanzees, and gorillas. Their overall size of their prefrontal cortex is about the same. And then we have humans way up here. You can see this is log scale. So this is actually more than 10 times the overall size in humans relative to the gorillas. Of course, it's important to take into account that brain sizes are different, right? Human brains are much bigger than macaques. So we have to account for that. And so we can look at this as a proportion. And so that's what these red and green dots are. These are two different studies that have been done. I presume maybe 2002 is a little bit more advanced. I don't know. So when you look proportionally, though, you see essentially the same pattern. That gibbons and macaques have a relatively larger percent of their brain dedicated to the prefrontal cortex, but they're about the same, right? It's around 35%. And that makes sense. You look at this, it's like, okay, yeah, 35% of the cortex. But when you do this for humans, chimpanzees, and gorillas, when you're looking at the overall size of the brain, so humans have a much bigger brain than chimpanzees and gorillas and orangutans, but in general, their overall proportion is approximately the same. So the prefrontal cortex proportion is approximately the same. It's just that you can see, so on the x-axis, this is where we have the overall volume, and you can see that humans just have a much bigger brain relative to gorillas, orangutans, and chimpanzees. The other thing to take into account is that gorillas, especially, are much bigger than humans' overall body size. So human to brain body ratio is much higher than the other apes. Remember, this is log scale, so you can see that the humans are, they got a brain that's nearly twice as big as, even bigger than chimpanzees, gorillas, and orangutans. We got big brains. But the percent is around 40% for all these animals. So this means that primates actually can serve as useful ways of looking at prefrontal cortex and their overall role. When we look at rodents, cats, other mammals that don't appear to have complex decision making, there's less to learn because these areas of their brain are relatively small. But primates have relatively large prefrontal cortices, and so a lot of experiments have been done. So we're going to talk about primate prefrontal cortex quite a bit, non-human primates. We're going to begin our exploration of the brain's function in decision making and reasoning by looking at the orbital prefrontal cortex. So this is towards the front of the brain, and its role in evaluation. Evaluation is defined as the estimation of a value of an option relative to other options based on past and present experience. We do evaluation all the time. You walk into the grocery store, one of the easiest things to estimate overall value is to just look at the price. So the grocery store is telling you things are worth this much. But sometimes, you're like looking at, say, the generic breakfast cereal versus the name brand cereal, and the generic cereal is cheaper, but does it taste better? Maybe it's kind of gross. So you might have to weigh those things: taste, presentation, and cost. You're doing evaluation at that moment. You might be comparing multiple breakfast cereals. You can have a ranking of things that are, this is really, really good, but this is super expensive. Here's a medium cereal, here's a generic cereal. You got to weigh all of these things. And the orbital prefrontal cortex plays a major role in this. Turns out primates do this very well, humans and non-human primates. And one way of doing this is to look at relative value of food. Monkeys are really no different. They like certain foods more than others. So for instance, monkeys will like candy. They will eat M&Ms. M&Ms is a great motivator for monkeys. They also like cherries. So they will eat cherries, and they'll prefer cherries over, say, peanuts, which is something they like less than cherries. And of course, a third choice might be a bowl of cucumbers, or in this case, a bowl of nothing, which clearly these two are more than this. So they can rank these things. And this is just sort of natural inherent experience. This monkey has had all these foods before, and it knows that it likes cherries more than it likes peanuts, more than it likes cucumbers, or say nothing. So this is based on past experience. And you can do a task to have them weigh relative values and how they choose things. This is known as the reinforcer devaluation task. So a monkey is trained with pairs of stimuli that are associated through training with either a reward or no reward. So in the training phase, the monkey learns that it has to make a pair in this case. So it's given a bunch of different objects. And through training, it'll learn that it has to, say, make these pairs and associate two stimuli. In this case, we're looking at different objects. This can also be different pictures, as we'll see in the next slide. So presenting them with different objects, and that they have to pair these two things. So the green cylinder with the yellow pyramid, if they make those associations that these two things go together, they get a cherry. Also in the training phase, you can give them different objects. And they'll learn that, say, okay, well, these two objects, I have to make an association with them in order to get a peanut. And so they will learn that, say, these two objects are associated with peanuts, these two objects are associated with cherries. And now when you give them the choice between a blue cube, which is associated with peanuts, and a yellow pyramid, which is associated with cherries, which one do you think they're going to choose? Well, in this case, the monkey is it'll evaluate his choices. He's learned that the pyramid is associated with cherries, so he's got to make that connection of, oh, this thing is connected to a reward that I really like, And this thing is connected to a reward that I'm not like super thrilled with. So the monkey's going to be more likely to pick cherries over peanuts, and therefore he's going to be more likely to pick the yellow pyramid because that's associated with cherries than the blue cube, which is associated with peanuts. So he's got to make that leap. So you're not just putting the food in front of him because that's too easy. You got to make this connection. There's got to be the training part of this, the evaluation, the relative value of these objects, leading to some sort of natural reward. So the monkey chooses between these two objects for cherries or peanuts. So let's look at results of what's happening in the orbital prefrontal cortex when a monkey undergoes this process. So keeping all that in mind that we just discussed about these objects that are associated with high reward, medium reward, low reward, now we're going to be looking at what's happening in the orbital prefrontal cortex and a monkey having to face these choices. But the order in which things are being presented is relatively complex, so bear with me as I walk through it. So the first phase is the instruction phase. So at this point, the monkey is going to be presented with two options. It'll be presented, say, with a yellow pyramid, which we have learned is associated with cherries, or a blue cube, which is associated with peanuts. The monkey has an inherent choice of preferring cherries over peanuts, and in this case, it has learned that the yellow pyramid is associated with cherries, the blue cube is associated with peanuts. So during that instruction phase, it's presented with both of these. And then the monkey is given a trigger. So it will then have the option of either picking, it'll be asked, you know, at this point, you can choose the yellow or not the yellow. So that is like one option. Or it will be presented with, you can choose the blue or not the blue. Okay, so it has one choice or the other. So that's one slight difference here. It's not able to really choose between. It's given the choice of picking the yellow or picking the blue. And then it can go back and pick it again. So in this case, it's given the instruction. So it's seeing, okay, I got yellow here, I got blue here. I definitely want the yellow, so I hope I get the yellow trigger. So as soon as it gets the yellow trigger, then it has an increase in activity in the orbital prefrontal cortex if it's given the yellow relative to blue. So the orbital prefrontal cortex starts to increase in its activity. Then it is given the option to open the box. So the box will have the reward inside. It can't see the reward, but there will be a box there that it can then open and then get the reward. And you can see the orbital prefrontal cortex then has activity as the monkey gets the reward. So the line is where the box is open. And you can see there's a bunch of anticipatory activity in the orbital prefrontal cortex when it is given the option to pick the yellow, because it knows yellow is going to give me cherries and it doesn't want any stinking peanuts. So in the peanut situation, when the trigger says, hey, you could pick a peanut or not, the monkey has no increase in activity. It doesn't really want that because it would prefer the cherries. It knows the cherries are better than peanuts. So the orbital prefrontal cortex doesn't have this increase in activity. But once it gets its reward, it's like, okay, well, I guess I'll eat some peanuts. But you can see the activity here is much less than here. So then they redo this experiment. Now comparing peanuts to, say, an empty bowl. I think in this experiment, actually, in this particular experiment, they use cucumbers. So peanuts relative to cucumbers. And so in this case, the monkey still knows that the blue cube is associated with peanuts. And so in the instruction phase here, it is shown, all right, you got the blue cube or this red cylinder, which is associated with a really low award. Now the blue cube, because it likes peanuts over, say, cucumbers or nothing, that it definitely wants the peanuts. And so now there's an increase in activity because it prefers peanuts over cucumbers. And then for the low reward, which in this case would be, say, cucumbers, I know it shows an empty bowl here, but bear with me, there is not that subsequent increase in activity. That it prefers the peanuts over cucumbers, and it looks a lot like in the previous case when the low reward was peanuts. So you can see that the interesting thing to compare here is these two graphs. When peanuts are considered the low reward, the activity is relatively low in the orbital prefrontal cortex because it definitely would prefer cherries. When it's considered the high reward, now it looks just like it did with cherries, and that's because the monkey prefers peanuts over cucumbers. And there's an increase in activity in the orbital prefrontal cortex associated with that. This is showing that activity in this area clearly is involved with evaluation. When given two choices, it's going to want to prefer the better choice, cherries over peanuts, or peanuts over nothing or cucumbers. Hopefully that's clear. When you lesion the orbital prefrontal cortex, it impairs monkeys' abilities to choose better food choices when it's done in this association with objects and food. So in this case, we had monkeys that were lesioned in the orbital prefrontal cortex. These in this graph, it's called PFO, so it's the orbital prefrontal cortex, or not. Those are the controls, CON. The monkeys were trained to associate 30 pairs of objects with either half a peanut or M&Ms in this case, in this experiment. In test 3, monkeys had to pick the correct object in order to get the M&Ms. So, and prefrontal cortex lesion monkeys had a very hard time picking M&M associated objects in test 3. So in test 3, they had the objects in order to get the food. And you can see the controls that they are given a score. The higher the score, the better they are at associating the object with the food. And you can see that all of these monkeys, each little shape here is an individual monkey, did much better when they were not lesioned versus the monkeys that were lesioned. You can see one of these monkeys couldn't do it at all. But when they were in test 4, they were just presented with food items. So they were given M&Ms and peanuts. These monkeys could definitely pick M&Ms. So when it's an easy driven case where they don't have to do any evaluation in thinking of associating objects, such as like associating money with an object or associating, having some different connection, and it's not just an inherent reward, lesions of the prefrontal cortex compromise their ability to make that association. But it's the food choice is something that's really basic and driven. And they can make that association themselves right away. So and that's because the food itself has the inherent reward. There's no really planning or thinking involved with this in associating those food choices. So we've talked about the orbital prefrontal cortex. That's located here. And I want to talk a little bit about the overall connectivity in the orbital with the prefrontal cortex to the rest of the brain. So we've talked about how the prefrontal cortex has to have sensory input, and the prefrontal cortex, in fact, receives input from all senses. And this gives this area the ability to evaluate all options: smell, taste, sight, sound. It has to evaluate the entire environment in order to do that. And so the prefrontal cortex will receive input from all these areas. There's also connections within the prefrontal cortex and with connections to areas outside of the prefrontal cortex. They are bidirectional, so many of these areas will connect to other areas. All these different lines are showing various connections. We kind of have sort of separate networks with the lateral prefrontal cortex receiving input from these areas, a lot of the visual areas and sensory areas. We've got the ventral medial and orbital frontal prefrontal cortex taking information from medial temporal lobe, which is important for memory, also visually driven memory, somatosensory cues, the amygdala, so emotional input. And then there's also all these areas get shared regions of connectivity from the basal ganglia, the thalamus, the insula. So there's a ton of connectivity. I don't expect you to have this memorized. What I'm trying to show is that when we're looking at circuits and connectivity within the prefrontal cortex, it actually becomes very complex because it's a highly interconnected area. Makes sense. In order for the prefrontal cortex to do what it needs to do, make these complex decisions based on all kinds of sensory input and previous experience, it has to be connected to all the other areas that we've learned about. So this is part of the reason why we're waiting till near the end of this course to talk about the prefrontal cortex, because it's basically involving all of these things. And yeah, I'm just mostly showing this so that you can have an appreciation for just how interconnected the prefrontal cortex it is with itself and with the rest of the brain. All right, the next part of the brain we're going to talk about is the dorsal lateral prefrontal cortex. So this is towards the top of the brain and towards the outside of the brain. So we're recording from that part, dorsal lateral prefrontal cortex. In this experiment, monkeys are trained to fixate their gaze on a central spot. So this is a visually guided task. Here's the FP, the fixation cross. And then while they are fixating, a saccade target appears in the periphery at one of 8 eccentric positions. So it's given a target. Monkeys then withhold a saccade. So they're told when they get this target, they're trained that they have to keep their eye on the target. They're not allowed to move their eyes to where the target is. They have to keep their eyes on the fixation point. Okay, so they got to keep their eyes there, but they can see in the periphery that one of these 8 spots has a target. So like a red square or something like that. Then the target disappears and the monkey is required to remember the position of the target. So this is like a short-term working memory before they're given some sort of go cue, like a tone that indicates the monkey should then shift its gaze to where the target was. Firing rates of individual neurons in the dorsal lateral prefrontal cortex depend upon the position of the remembered cue, meaning they carry sufficient information to store the short-term memory. And this change is observable in averages of the firing rate for each position, and critically, is observed during the delay phase of the period, when there is no information on the screen, and short-term memory is the only site of information maintenance. Okay, so there's a lot there. Let me walk through what this precisely means. So first of all, this portion of the dorsolateral prefrontal cortex, what we're seeing is that it has a certain preference of location. So this is 135 degrees here. So this is this spot here. You can see that most of the activity is happening there. But it also happens a little bit at 90 degrees and 180 degrees. As we learned during the the way the superior colliculus works, is that there tends to be preferences for places within the visual field. So this is a visually related area of the brain. The thing that's crazy about this, though, is that during this presentation part, there's a fixation point, right? They have to fixate. They are given a target, right? So a target appears in this phase, and then it disappears. This is the phase when the target shows up. And then it disappears. And there's nothing there. It's just like it was before. So this is with no target. Now there's target. And as soon as it disappears, there's an increase in the activity of the dorsolateral prefrontal cortex. What the monkey is seeing here is exactly what it's seeing here. There's nothing different. This is different because there's a target there. But as soon as it disappears, now there's an increase in activity. So this area of the brain is saying, oh, okay, I know I need to do something. I'm going to have to move my eyes to this area. So this is like a pre-planning phase, and it's maintaining a short-term memory so that it can make a response. And then as soon as it's given the go signal, so that's what this line is, now the activity decreases. So this is the most important figure here. You can see this is a raster plot. We're looking at histograms here. And during the delay phase is when we see this increase. And again, remember, the crazy thing about this is that what the monkey sees in the screen during the delay phase is identical to this phase here. It's just that it was given a stimulus before, and then it disappeared, and it had to remember where it was located. And so this area is maintaining that memory, allowing them to make a choice. If the monkey is not trained to do anything like this, it doesn't actually have this. So it has to learn that it's going to get some sort of reward. So this is essentially planning for a visually guided task. So it knows that it needs to do something, and it has to maintain that memory in order to get a reward during the response when it will move to the location. You can see other areas. So the opposite area here, we don't see it because this is dependent upon the visual field, like where is actually happening in the visual field. So this is a combination of things that we have already learned, that there's a visual field in front of us. Certain areas of the visual field will be sensitive. Certain parts of the brain will be sensitive to certain areas of the visual field. but what's actually happening here is what was in the visual field, and that there's a task associated with it where you're going to get some kind of reward. Another experiment which has some really interesting results for data collected from the prefrontal cortex involves the match-non-match test. So I'm going to walk through this test first because it's kind of complex, and then we'll talk about the neural data and what actually happens in the brain. So in this case, the monkey is going to have not two choices, but two rules that it'll have to follow. One is the match rule. So let's walk through that one first. So a monkey will be given a picture. It'll be shown a picture such as a sailboat. And it will also be given a cue, which will tell that monkey, hey, you need to follow the match rule, which is the next time you see this image, you will need to release a lever. So the monkey's holding on to this lever, and it'll let go of the lever if both objects match. So in this case, it's given a juice drop. It could be a picture, it could be a sound. Through training, the monkey will learn that it has to follow the match rule. So in this case, it's getting a juice drop. And so when it gets a little tiny bit of juice, it says, oh, okay, I have to follow the match rule, which is look for this picture again. So then there's a little delay phase. And then the monkey is given one of two options. It might get this picture, and sometimes it might get a picture that does not match. If it sees the picture that matches, it is supposed to respond. It is supposed to release the lever. So if it saw this object or this picture, then it's going to, it knows that it's following the match rule because it got the juice drop. And so the monkey, if it does it correctly, he will release the lever. But if he sees this, a light bulb, this does not match this. And so the monkey is supposed to continue to hold the lever. If he releases the lever, he messed up the experiment. If it's this, then the monkey will be given another delay, and then it'll be shown another image that will be the match, and then the monkey will need to respond then. So it's got to look for the boat, right, as the match. And then in the next time, it might be the light bulb is the thing that it has to match, and that the boat will be the non-match, right? Or some other kind of image. So the monkeys are given a whole bunch of images. It's not image dependent. It's not like they're learning, oh, I always have to match boats. What they have to do is they have to remember what the rule is. They're given a cue for what the rule is, and then they got to follow the rule, whatever the image is. It might be a boat. It might be a light bulb. It might be Chef from South Park. And I'm not kidding. In this experiment, this is actually the image taken from the paper where they describe this. This was published in Nature, and they did use Chef from South Park that they showed to these monkeys. So this is actually their image. All right, so that's the match rule. Then there's the non-match rule. So in this case, it'll be some kind of different cue which will inform the monkey that they're supposed to follow the non-match rule. So now when they're given an image, say like in this case a green background, it could be a tone, it could be something else, some kind of sensory modality that isn't specific to, like, say, visual cue. It might also be an auditory cue. It might be a taste cue. So if they're given the cue that the monkey has learned is like, okay, I have to follow the non-match rule, there'll be a delay phase. And now they're given one of two images. And the correct response will be if the image does not match the previous image during when they were told that they're following the non-match rule. So in this case, the monkey needs to then release the lever. If the image matches, this is the wrong cue. They're following the non-match, right? So if it's the same image, they have to continue to hold the lever, and then there's a delay, and then the next image will be the non-match, and then they should respond, right? So you can see it's kind of the opposite of this. I mean, it is the opposite, right? In this case, they're looking for the match, and in this case, they're looking for the non-match. It's not image dependent. It can change. The image can change from trial to trial. What it's dependent upon is what rule they're following, and they're given the rule by the kind of stimulus they get with this first sample phase. Hopefully that's clear. Replay this again if it's not. In this figure, what we're looking at is data from one single dorsolateral prefrontal cortex neuron that showed an increase in the activity of the delay phase during the match test. So let me walk through this. So we got two kinds of tests, right? We got the match and we got non-match. We've got sample and we've also got delay, right? So we got these two phases and we're looking at neural activity across the whole thing. Like I said, the monkeys have two different stimuli that they get to tell them that there's the match rule. Two different kinds of sensory stimulus. One is taste. So if they get a little bit of juice or if they get a low tone, that is going to tell the monkey, okay, I need to follow the match rule. If they get no juice or a high tone, this is going to tell them that they need to follow the non-match rule. Or it could be, say, like a background color or something like that, right? You can do different things here. In this case, they did no juice, high tone, juice, low tone. So it'll be one of those two cues, two different kinds of sensory modalities. So it's not dependent upon like, oh, I got juice or I didn't get juice. It also is tone, like low tone or high tone. They have to distinguish those things as well. In this one neuron that they were looking at, the neuron increased its activity during the delay phase. So you can see the firing rate increased for blue and green. This is when they're following the match rule during this phase. It didn't increase during the sample phase. It just increased during the delay phase. And it didn't matter what picture they were looking at, whether it was, say, a boat, a light bulb, Chef from South Park, or, I don't know, picture of a bicycle or something like that. The neuron increases its activity during the delay phase, and it didn't matter if it got juice as the cue or low tone as the cue. It was just following the match rule. And during the delay phase, when there's nothing there, this neuron is increasing its activity because it knows it needs to follow the match rule. So that's this one neuron, but other neurons responded in different ways. So let's look at that. So we're going to look at the summary of all the data from this experiment. So neurons were either selective for either match or non-match conditions. So that's the first part. And then some neurons had high response during the sample phase, and other neurons had high response during the delay phase. So let's look through what this histogram shows. We can evaluate the activity of a neuron, how it responds relative to baseline, to see whether it prefers non-match or match. So the neuron that we saw before, it would actually fall in this category. So it increased during the delay phase, and it would be one of these neurons, one of these cells where it increased activity during match rules, not non-match rules. There were other neurons that increased activity during non-match rules and not match rules. There were neurons that increased their activity, did the same thing, but during the sample phase, either match or non-match. So they had neurons, so they looked at different neurons, right? You can count up the neurons here, so number of neurons here. This is 10, this is like 12. They looked at a bunch of neurons, like hundreds of neurons here. And they fell into one of these four categories. They were either, during the sample phase, they were either match or non-match, nothing really in between, right? There wasn't any one neuron that just responded to both. Or they responded to match or non-match during the delay phase. So they have neurons that, so what I'm trying to say is that the dorsal lateral prefrontal cortex has complex response properties depending upon which neuron you're looking at. And so any of these neurons are going to be active depending upon the conditions. And it's going to be helping the monkey make those decisions of, oh, okay, neurons that are active here during the delay phase, during the match phase, these neurons are going to be helping those monkeys maintain that choice. And then different neurons are going to help the monkey make their correct choice, say during non-match during the sample phase. And so you can see that it's a relatively complex network. Hopefully you can appreciate what I'm talking about here. So there are other tasks that we're going to talk about. So in the prefrontal cortex, we're going to talk about attention and working memory. So we've already talked about working memory a little bit, but we're going to talk about it in the context of the Wisconsin card sort task. So prefrontal cortical areas, we know that they mediate planning and decision making and helps to coordinate direct attentional processes. We talked about how the prefrontal cortex has changes in activity associated with working memory. You know, the delay phase when monkeys are shown some sort of reward between two cups, and then they get a delay, and there's activity during that delay, right? We know that the prefrontal cortex can be assessed by various neuropsychological tests in humans, such as this task. So in this task, a subject is given a deck of cards with different shapes, different numbers of shapes, and different colors. And the subject is supposed to start sorting the cards. The subject chooses an initial sorting strategy. So they just have to pick. So let's say they're going to pick shape. And they're told whether they are right or wrong. So they're not told what the rule is. They got to figure out what the rule is. So as they're sorting, so like, you know, putting stars here, and then maybe this person's like, okay, well, maybe I need to sort stars. So I'll put this one also here. And when they make a mistake, then they're told, oh, nope, that's wrong. So the mistake then is like, okay, well, I guess I'm not sorting by shape. I guess I have to sort by number or color. So then they have to redo it and then try to figure out the rule. And they're only told whether they're right or wrong. And then, so they're doing this, and then they kind of figure it out. And then all of a sudden, the rules change. They're not told when the rules change, but they have to perceive that the rules have changed. And now they're told that they're making errors. Now they have to learn the correct course by changing a different strategy. So this is a relatively complex task. So people who have prefrontal cortical damage, they have difficulty switching their cognitive plan and will continue to make wrong choices. These people have a tough time changing course. There's also something known as the STROOP task. You may have seen this. This is a popular psychological type of experiment. It involves the frontal association cortex. So the frontal association cortex, it mediates planning and decision making and helping to coordinate directional attentional processes. So in the STROOP task, a subject is asked to attend to salient features of a stimulus in spite of distraction. So in order to successfully complete this task, the subject has to pay attention to the instruction. So the first part of this is really easy. We just go through here and we have to say the number of the color that you see. So this is dark gray, light gray, brown, pink, green, blue. You just go through, pretty easy. All right, because there's only one thing that you have to attend. Now let's change it up. You have to say the name of the color of each word. So what do we mean here? So now when we're doing this, this can be much harder. So this is the STROOP task. So either you have to say the name of the color of the letters, or you have to read the letter and read the letters and pay attention to what the word is saying. So in that case, it would be red, green, blue, pink, brown, orange, green, pink, red. But if you have to say the name of the colors of the letters, then you have to say light blue. This is hard. Dark gray, red, brown, pink, green, purple, brown. This is much harder when you have to flip. Okay, so you have to pay attention to these things. So let's talk about what happens then in the brain. When you do repeated trials with the STROOP test, interestingly, you can see that the activity in the anterior cingulate cortex, so this is on the medial side of the brain towards the front, the part of the cingulate cortex, that activity is greatly enhanced. This is during phases when there's cognitive conflict. So when you're doing that STROOP task, you're struggling very hard to decide what you need to say. Do I need to say green? Do I need to say brown here? Really kind of hard. And because you're trying to remember whatever the instruction was, but your instinct might be to look and just read the word. So this, okay, let me walk through this figure here because it's relatively complex. When activity in the ACC is high, so this is during fMRI, and we're looking at activity in the ACC. When activity in the ACC is high, this will predict activity in the lateral prefrontal cortex in the next response, in the next phase. So following conflicting trials, individuals, let me just read this through and I'll just repeat what I said. Following conflicting trials, individuals adjust their behavior to improve their performance. This adjustment is thought to reflect changes driven by inputs to the dorsal lateral prefrontal cortex from the ACC. Activation of the ACC on the previous trial as measured by fMRI predicts the response of the dorsal lateral prefrontal cortex on the current trial, and this predicts adjustment. So let's walk through this again, what this is showing. So when a subject is struggling with a choice and it's learning how to do the STROOP task, you're going to see increased activity in the ACC. In the next trial, in the very next choice that the subject makes, this is associated with increased activity in the lateral prefrontal cortex. So the ACC, whatever happened in the previous trial, is informing how the lateral prefrontal cortex needs to be paying attention. When there's lower activity in the ACC, so presumably the subject is not struggling too much, this predicts that activity in the lateral prefrontal cortex will be lower than average. So along this axis, this isn't time, right? What we're looking at is changes in ACC in the previous trial is associated with current activity in the lateral prefrontal cortex. So it predicts what we're going to see in the lateral prefrontal cortex. So what this is showing is that the ACC, as you're struggling to do the task, it's leading to changes in the very next trial in activity in the lateral prefrontal cortex. And this is going to allow the subjects to do better on this task over time. Another card task is the Iowa gambling task. And so one way to do this is that you can have certain decks that will, so the subjects, they're asked to make bets about like what card they're going to see, and the certain decks will give them, the odds will be very small wins and occasionally large losses. So they're going to get quite a few little wins, but occasionally large losses. When they set this up, over time, the subjects are supposed to learn that this is disadvantageous. So these frequent small wins, like you get 25 cents, 25 cents, 25 cents, but then you might lose $10. They're supposed to learn that these two decks are not working to their advantage, that they're losing a lot of money. In C and D, in this task, in the Iowa gambling task, they're getting frequent small losses. So you're losing 25 cents, 25 cents, 25 cents, but you get an occasional large win, like $10. And you're like, oh, okay, over time, you're going to learn that these decks are actually going to be paying off better, even though you're getting frequent small losses. So even though you're losing more often, you're going to end up on the plus side because of these large wins. So that's what C and D would be showing. In A and B, it's the opposite. You get frequent small wins, so it feels like, oh, okay, I'm winning, but it's the big losses that actually make these decks disadvantageous. So the subjects have to kind of play with the cards and figure out which is which. And they got to make choices about which one to exploit and like which one they're going to pay attention to. So most individuals start by preferring decks A and B because like you're getting these little wins. You're like, okay, yeah, I'm winning. But they eventually figure out that, you know what, actually I'm losing because of these big losses. So I'm not going to be using A and B, and instead they need to focus on C and D. Individuals who have lesions, so these are humans with lesions to the ventral medial prefrontal cortex, they generally do not make the transition to choosing the advantageous deck over the disadvantageous deck. So normal controls, over time, you can see that they're preferring the C and D deck to the A and B deck. They know that over time, they've seen that like, you know what, I'm not making as much money here with these two decks as I am with these. So then they start to use C and D. This is what normal controls will do. They started with A and B because they got those little wins because they thought like, oh, okay, I'm doing pretty good here. But then they see the big losses and they're like, no, I got to change my strategy. I'm going to use C and D. Even though I get more frequent losses here, I'm going to end up with more money because of the big wins. Individuals with lesions to this area of the brain, they never make this choice. They continue to exploit the A and B, and they take these big losses because they're getting those small frequent wins. They can't evaluate that, hey, you know what, actually this isn't a good choice. That in fact, what I'm ending up here is they end up with bigger losses. So they can't make that evaluation and that judgment. Now we're going to talk about the relationship of the insula to behavioral choices. So if you remember, the insula was that part of the brain that's hidden behind the temporal cortex. When you pull it away, you can see the insula hiding behind there. In this study, we're looking at humans. 19 subjects had insula damage, and but not damage to other parts of the brain, whereas 50 others had damage to other parts of the brain, but not damage to the insula. So these individuals served as a control group. All of these subjects were also smokers, so that we're looking for the relationship of brain damage to smoking. 32 of the total group had tried quitting. 37 of the group did not try. Those were the non-quitters. So they didn't even try to quit. For those that tried to quit, half of them were able to disrupt their smoking addiction. That is, that they were quitting smoking was relatively effortless, and it wasn't a big deal for them to quit. Whereas the other half found that they were unable to quit. It was really hard to quit smoking. So let's look through the data. So in the non-insula group, so this pie graph here totals 50. 31 of the 37 non-quitters, so people who couldn't even try to quit, were non-insula damaged. And of the people who tried to quit but failed to quit, 15 of those showed no disruption. Only 4 out of 50 were actually able to easily quit smoking. 4 out of 50. For those individuals who had total insula damage, 12 of the 19 subjects were able to quit relatively easily. And there appeared to be sort of a bias to the right insula versus the left insula, whereas almost all of the subjects who had right insula damage were able to quit, but left insula was also able to quit pretty well as well. More than half, and certainly a lot better than non-insula damage. So the conclusion of this is that subjects who experience disrupted smoking addiction were more likely to be insula lesion patients. This indicates that the insula is involved with some of the habit-forming processes around smoking, and that when individuals have insula damage, they have an easier time to quit. And so the insula is providing some of that overall habit-forming signal that makes it very difficult to quit smoking. And when you have damage to the insula, some of those previous experiences that might be stored by the insula and that overall basic association isn't there. The drive isn't there, so it's just easier to. I also want to talk a little bit about a super interesting feature of the brain, which is called the default mode network. So this is called the default mode function as well. The posterior cingulate cortex, so this is towards the back of the brain of the cingulate cortex, right? Posterior. This is the anterior cingulate cortex here. This is the posterior cingulate cortex. This is part of the default mode network. So let me walk through this. Images of the human brain showing metabolic responses as measured by PET scanning. So this is kind of like fMRI, but instead of looking at changes in blood flow, it's just looking at changes in overall metabolic activity. Okay, but essentially the same thing. Individuals were performing either standard laboratory tasks, certain cognitive tasks, or they viewed the same stimuli passively. So that's without any sort of task requirements. So they were either asked to do something or not. But they were shown either the stimuli or not. And the active task performance led to consistent decreases in activity in the default mode network, a set of regions whose responses are enhanced at cognitive rest. So a little bit about the default mode network. These areas, the posterior cingulate cortex, the dorsal medial cortex, the ventral medial prefrontal cortex, and certain parts of the posterior lateral cortices. These areas tend to be increased in activity when the brain is at rest. The way we identified the default mode network is actually a super interesting story. So subjects would be put into an fMRI machine, and they'd be told, okay, we're going to do a certain test here, but I need to get a baseline. So I want you to just not do anything. I just want you to just sit there. We're going to get a baseline of activity. And they would see activity in these brain areas. And they'd tell the subject, okay, well, you're doing something, so just don't do anything. Like, whatever you're doing, don't do it. And they'd be like, I'm not doing anything. And they would see activity in these brain areas. So this is like activity when we are not focused on a particular job or task. We see activity in these areas. And it's thought that activity in these areas is associated with our overall internal dialogue. When you're just sitting there and you're left with your own thoughts, you're going to be thinking about things. Like, even if you can't do these, if you're told to not do something, it's really hard to just shut off your brain completely. And that's the default mode network. It tends to be active during those phases. But when you're doing something active, such as looking at images and doing something that you have to do, you see a decrease in these activity areas. So the overall dialogue and reasoning about what you're looking at and thinking about what you're looking at, that shuts down because you're focused on a task. So these areas actually shut down. So prominent among these regions, as I said, is the PCC, the ventromedial prefrontal cortex, and certain posterior lateral cortices. Right? So the default mode network, the resting metabolic rates in human brain they're actually really high in the default mode network. In fact, it's especially high in the posterior cingulate cortex, which is among the most metabolically active regions of the brain. So these areas of the brain, despite the fact that they actually turn down their activity when we are doing some sort of active task that requires focus, these areas are in fact some of the most metabolically active areas of the brain. And just kind of showing that, okay, these areas are actually pretty critical and they're doing an important brain function. This pattern of activity is also seen in monkeys. So this is data taken from, I believe this was done in macaques, looking at activity in the posterior cingulate cortex. So this is firing rate, spikes per second, looking at this very high metabolic area that's involved with the whole dialogue process. So in this case, monkeys were put into 3 different conditions. They were either given no task whatsoever, or they were given some sort of task where they had to pay attention to something and make choices about what they're perceiving, or they were given some kind of memory task where they had to recall something that they had learned before. What they see is that they see sustained reductions in activity during the cognitively demanding parts of the task. And so this area of the brain, just like it does in humans, when they're focused on some sort of active task that's relatively difficult and they got to pay attention, activity decreases. So tonic firing rates during the delay were reduced during the difficult part of the task relative to the rest condition. And when information was a help in short-term memory, responses were further reduced. But during no task, activity was high. So with no task, the activity is high because the monkey is presumably letting its mind wander a little bit and thinking about things. But when it needs to focus, activity in the PCC actually decreases its activity. One way of looking at the role of the posterior cingulate cortex and presumably the default mode network is by using what's called the forearm banded task. And this is testing exploratory behavior. So exploratory behavior in animals, this of course includes monkeys. This can be studied in this task. And in this task, they're given 4 options to choose from. And each option gives a random reward. So in this case, some juice. Some of the options give more juice than others. Okay, so in this case, it picks pink, and it's going to get, say, a certain amount of a reward. Each option gives some juice, but the monkey wants to pick the one that gives it the most juice because it likes juice. Give me that juice, baby. And so, yeah, the monkey's got to explore. It's got to figure out what the rule is. Like, oh, okay, I guess right now red is giving me a lot of juice. But then it changes from time to time. It changes a little bit. And so maybe the next time pink will actually give you more juice. So they have to explore. There are times when it figures it out. Okay, red is the way to go. So I'm just going to take this, and it will exploit the rule. But it does need to occasionally change its choice to see maybe one of these options is actually better. This is exploratory behavior. So this is actually kind of a complex task. It's not like, oh, pink always gives me a reward. None of these other ones give me a reward. It has to evaluate and test. Maybe yellow is the thing that's giving me a good reward. Red was doing it before, but maybe yellow is better. So it has to explore and change its choices. So there's a lot of thinking involved with this test. So let's look at what happens just for the overall choices. All right, in this graph, lines in this panel indicate the value that is offered for each option. So this is just showing that initially we all start at the same point. All the different options have the same amount, but now the amount that each option offers changes. So yellow and blue is getting better, whereas red and pink is getting worse. But now pink is better. You can see it's just randomly changing. The star indicates the choice that the monkey made. So the first option that it picked was yellow, and it got some juice. But then it was like, okay, well, that was some juice. Maybe I'll try red. Red gave me a little bit less. Okay, I didn't actually benefit from that. So let's try yellow again. Oh, yellow gives me more juice. I'm going to stick with yellow for a little bit. Let's check blue. Blue, nah, not so great. Maybe we'll try red. Red, oh, it's increasing a little bit, but maybe I'll try yellow, and yellow is better. And now, because it's now seen yellow giving more juice compared to blue and red, now it's going to exploit. It's going to say, you know what? Yep, I'm getting a lot of juice with yellow. I've tried the other options. Yellow is the way to go. And so now it just chooses yellow, 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 because it just changes a little bit each time. And then eventually, yellow starts to go down because of random changes. And now it's like, you know what? Maybe I don't want yellow. Maybe I'll try pink. Maybe I'll try red again. Well, let's go back to yellow because pink wasn't much better than yellow. No, pink wasn't so great. Now yellow is actually better, but let's try red and blue. Nah, actually yellow is the way to go, so I'm going to exploit yellow again. So now I'm going to follow this rule. So there's portions of the test where it has to explore. That's what's happening here in this phase. This is an exploratory phase. It's trying to figure out which option is best. It eventually settles on yellow, and now it's following a rule. It says, you know what? Yellow is the way to go. I'm just going to always pick yellow because I'm getting lots of juice. But eventually, it starts to give less juice because of random changes, and now it needs to explore again. So this would be an exploratory phase where it's trying its different options. Eventually, it's back to yellow. Yellow is the way to go, so it's going to exploit during this phase. It's just going to follow a basic rule. So we have two phases during this test, and we can tell what the monkey is doing by its choices. If it chooses yellow or some other color over and over and over again, it's exploiting a rule. It's getting the most juice. When that rule starts to break down, it needs to switch over and do an exploratory phase where it has to think through its options. It's got to be like, was blue better than yellow? Was pink better than blue? I got to figure this out. Eventually, once it finds, no, yellow is the way to go, then it goes back to an exploitive phase again, okay? So please read through this. This explains it, basically what I just said. So there's some really interesting things happening in the default mode network, in particular the posterior cingulate cortex. The posterior cingulate cortex, so this is this area of the brain that we said, okay, it decreases its activity when animals are following some sort of active approach, when it's doing a memory task or it's doing some sort of active choice task, right? Like if it's following certain kinds of rules, it decreases its activity. We can look at its overall activity when it's either in the explore phase, which is in black, or in the exploitive phase when it's in red. Remember, when it's exploiting its options, it's picking yellow over and over and over again. It's following a basic rule. You can see that activity in the PCC is lower when it's exploiting and following this rule. So it's just basically following this memory task. than it is during the explore phase when the monkey is doing some active evaluation and it's thinking about, is red better than blue? Was that good? Maybe I want yellow again. I'm not too sure. So the PCC is actually, it's not like it's just shut down all the time. It actually is important when we are doing active evaluation and we're trying to figure out relative thinking and logicking of trying to figure out different rules. And when we are having some sort of internal dialogue. So these data suggest that the PCC, and obviously by extension, the default mode network, because the PCC is interconnected with other areas of the brain in the default mode network, that it regulates active reasoning and puzzling, and perhaps this is providing some sort of internal dialogue. So when the monkey is during this explore phase and it's weighing its different options, that's when the default mode network and the PCC is active. When it's following a basic rule, yellow is the way to go. I'm going to get a lot of juice with yellow. That's exploiting. That's the exploitation phase. It doesn't really need to think much because it's just getting a lot of juice. And activity in the PCC decreases because it's just following a basic rule and it's a simple task. Really cool stuff. So that's it for this lecture. Here's some key questions about decision making and planning. So first what are some of the key areas that regulate decision making and planning? What is the relative size of the prefrontal cortex in humans relative to other primates and other animals, mammals? What is the apparent role of the orbital prefrontal cortex? And what are the lines of evidence that support this conclusion? How do neurons in the dorsolateral prefrontal cortex change their activity during complex memory and planning tasks? How does activity in the anterior cingulate cortex affect activity in the dorsolateral prefrontal cortex? What is the effect of the insula on quitting smoking? And how does activity in the posterior cingulate cortex and the default mode network change during exploratory trials? So yeah, you can see why we waited on this, because it's a lot of complex behavioral and cognitive tasks, and it involves most areas of the brain. And this is where a lot of the complex things are happening within the brain, taking sensory input, providing motor output, but this is the part that's doing the evaluation of all that and allowing the animals and humans to make choices that leads to changes in behavior. So that's it for this lecture. We'll see you next time.